The shadow looks great patient separate into clear clusters on the March 27, 2026 plot. But the shadow captured only some of the information. How much exactly? And what do the flashlight angles actually mean in terms of your measurements? The spread of the shadows is the variance. More spread means more information preserved in the projection. PC captures the most variance, the widest shadow spread. DE captures less. P to even less. Plot the variance captured by each PC as a bar chart, a Scree plot. The bars decrease. At some point they flatten, the remaining PCs capture only noise. The elbow of the Scree plot tells you, stop here. Components above the elbow are signal. Components below are noise. Each PC is a direction, a flashlight angle, in the original 50-dimensional space. Mathematically, this direction is an eigenvector of the covariance matrix. But practically, it's a recipe. PC equals 0.4 times gene A plus 0.3 times gene B minus 0.1 times gene C, thus tiny contributions from the other 47 genes. These weights, the loadings, tell you which original measurements contribute most to that shadow direction. The flashlight angle is a weighted combination of all measurements. Loadings connect the abstract PC direction to actual biology. If gene A has a loading of 0.4 on PC, gene A strongly influences the first principal component. Patients with high gene A expression tend to be on the right side of the PC shadow. If gene AC has a loading of 0.001, gene AC is irrelevant to PC, it doesn't contribute to the shadow's spread. Plotting loadings reveals which genes drive the separation you see in the PCA scatter plot. The answer is in the loadings. PC captures 45% of the total variance in the data. PC captures 25%. Together, 70%. Add PC at 15%, 85%. 3 shadow dimensions capture 85% of the information from 50 original dimensions. The other 47 dimensions collectively contribute only 15%, mostly noise. The cumulative variance plot shows this, a curve that rises steeply at first, then flattens. When the curve reaches 90%, you know how many PCs you need. In cancer genomics, 20,000 genes measured across 200 tumor samples. PCA reduces to 2 dimensions. On the scatter plot, breast cancer samples cluster on the left, lung cancer on the right. This is separated tissue types. The loadings of PC reveal the genes with the highest loadings are known tissue-specific transcription factors. They have found the biology automatically. In clinical data, 50 patient features labs, vitals, demographics. They shows 3 clusters. The loadings reveal PC is driven by kidney function markers, PC by inflammatory markers. Scree PCs capture 90% of the clinical variation. Gather marbles on the table your data in high-dimensional space. Shine the flashlight, PCA finds the optimal projection axis. The shadow on the wall, the low-dimensional representation that preserves maximum variance. The angle, the eigenvector with a loading recipe. The spread, the eigenvalue measuring captured information. 50 dimensions become 2 meaningful shadows. PCA found the story hiding in the noise.